Okay, so let's look at a nerve impulse. A nerve impulse is also known as an action potential. So before we get started on this, what I want you guys to know is that if we take in sodium chloride, so just regular table salt, sodium chloride doesn't stay sodium chloride. It will separate in water into sodium ions and chloride ions. And the same thing happens when we take in another salt called potassium chloride. So it too will separate into potassium ions and chloride ions. So since we're mostly water, when we take in those salts, they separate into ions, and that's how they are in our body. All right? And these two ions, sodium ions, shown here in pink, and potassium ions, shown here in purple, are going to be very important in a nerve impulse. So let's start off with the normal situation where we don't have an impulse moving along a membrane. And so this is what is known as resting potential. So resting potential is the difference in electrical charge between the inside and the outside of an undisturbed nerve cell. So what happens here is the plasma membrane is polarized, meaning it's positive on one side and it's negative on the other. So let's look at outside the plasma membrane. Outside the plasma membrane, we find a large number of sodium ions there, okay? Very few potassium ions. So because of all these positively charges found outside of the cell, the outside of the plasma membrane of a nerve cell is positive. Inside the plasma membrane, we have a lot of potassium ions, very few sodium ions. But what we also have inside of a neuron are a large number of negatively charged proteins. And so because there's a large number of negatively charged proteins, this makes inside of the plasma membrane negative. Now to maintain this, we use this thing called the sodium-potassium pump. And so the sodium-potassium pump is a carrier protein, and it does active transport. And what it constantly does is it constantly pumps sodium out of the cell and potassium into the cell. So this is one of the reasons why nerve cells have a high metabolic rate. It's because this pump is working all the time. Okay? So before we can have an impulse occur, we have to get above what is known as the threshold potential. And the threshold potential is a stimulation level that must be exceeded to cause a nerve impulse. Okay? So, you know, if you've ever looked down on your leg and you see a mosquito feeding upon you and you're like, I didn't even feel that land. Well, it landed below threshold, all right? So if it lands, you know, unlucky for the mosquito, lucky for you, it lands on the end of a nerve ending, you know, it can be above threshold then, and you feel it when it lands. Okay, so let's look at an action potential here, okay? So this is showing this here, sodium out, potassium in, positive out, negative in, all right? So the first thing to happen here is what is known as depolarization. And this is a loss of electrical charge on the surface of a membrane, okay? So if we get an impulse above threshold, what's gonna happen here is these sodium ion channels specific to sodium, are gonna open up. And this is gonna allow sodium to diffuse into the cell. So let's go back here at resting. At rest potential, sodium out, potassium in, positive out, negative in. So what we have now is we get this influx of positive ions into the cell. Was positive here, negative here. If we get a lot of positively charged ions going into a negative area, we have now neutralized that area right there. Okay? So that's that depolarization, loss of electrical charge. No charge on the membrane. Now, this is going to be quickly followed by repolarization. So repolarization is the movement of the membrane potential to its initial resting state, meaning that it's polarized again, all right? But it's going to be polarized in a different way. So here, when sodium ion diffusion slows down, the potassium ion channels open, and this allows potassium ions to simply diffuse out of the cell, okay? So, allows potassium to diffuse out of the cell. So this area was neutral. But we're taking positive ions and putting it into a neutral area. So this area becomes positive again. And we're removing positive ions from a neutral area, so that area becomes negative again. So we get that repolarization. Now the plasma membrane is polarized again. Okay? So these changes in charge across the plasma membrane are going to power an impulse. So an action potential or nerve impulse is a rapid sequence of depolarization and repolarization along a nerve cell membrane. We go from having a polarized membrane, sodium out, potassium in, to a neutral membrane, sodium in now, neutralized membrane, to a repolarized membrane, potassium out now, sodium in, positive out, negative in again. Okay? So the way to get this back to return to the resting potential is for that sodium-potassium pump to work again. Okay? So the sodium-potassium pump restores this back to its original resting potential. There's a small refractory period where an impulse cannot occur there. So propagation of a nerve impulse. So how we get a nerve impulse to move down along an axon or a nerve cell is that an action potential in one area is going to cause action potentials to occur in adjacent areas. So kind of like a domino effect. If it occurs here, it's going to occur there, then it occurs there, and so on and so on. Now typically impulses move in one direction because the impulse would start here on a dendrite or the cell body, and from there it would move down the axon. Okay? Now if you prodded this in the middle of the axon, it would move bidirectionally, but that rarely occurs. Okay? Now, let's look at the function of myelin, how myelin influences this. Okay? So, myelin, so if we look at unmyelinated areas, and this is pretty much what I just kind of talked about, or unmyelinated areas. Unmyelinated areas are going to conduct impulses over the entire plasma membrane surface. So if the impulse starts here in the dendrites, then it's going to move through the cell body and then down the axon. So here to here to here to here, and so on. Okay? Myelin, what myelin is an insulator, and it does not allow an impulse along that area where it's found. Okay? But, we're not done with this here. So if we go to a similar kind of picture here we got Schwann cells along the axon, and we can see these spaces in between the Schwann cells. So the Schwann cells is where we have myelin. This area where we don't have myelin is known as a node of Ranvier. It's an unmyelinated area. And those small unmyelinated areas can conduct an impulse. So what happens here is that when we have an impulse, it's not going to go from this area to this area to this area to this area, which is what happened in an unmyelinated area. That impulse will move from node to node. So it'll go from here to here to here to here. All right? And so by doing that, instead of having to travel through this area right here, that impulse skips to there, and skips to there, and skips to there. And by skipping over these myelinated areas, it's going to move much faster. Okay? So the same thing happens. Sodium diffuses in those areas. Now, I do want to point out, these nodes of Ranvier are tiny. So this is showing between two Schwann cells. You see that little line? That's a node of Ranvier. Okay? So even on these pictures, they make these node of Ranviers much, much larger than what they actually are. So this moves that impulse much, much faster by having these myelinated, by having Schwann cells or oligodendrocytes there. And so that's why white matter is where we do the conduction. We're sending impulses from one body, part of our brain or spinal cord to another. Okay? Last thing I want to talk about is all or none response. In here, a neuron responds completely or not at all to stimulation. So it's either we get above threshold, we have an impulse, or we don't. Okay? If we have a greater intensity of stimulation, right, so something's more painful or more pleasurable, right, or tastes better, right, we're going to get more impulses per second. So, we get a higher frequency.